I wanted to talk about the lessons from reference-free exRNA deconvolution by the exDec deconvolution algorithm. But before we dive into exRNA, I'd like to take a little broader picture and include tissue-based deconvolution, something that we've just heard about. Within tissues, the issue is cell type composition and bulk profiles, whether RNA-seq and DNA methylation need to be deconvoluted to get at the cell type proportions and cell type specific states. With biofluids, it's not the cells, it's exRNA carriers, vesicles, lipoproteins, RNP particles. But the readout is similar, RNA-seq. So we developed exDec with an idea that we can do both tissue and biofluid deconvolution, building on our previously developed epigenomic deconvolution method called EDAC. So as you can see, EDAC is published in 2016. It's one of the, I just counted, one of the 40 papers or something on deconvolution, or 20 to 40, I forgot exactly from the previous talk how many of them appeared in that year. We've been refining it ever since and applying it in different contexts, both tumor deconvolution context, where we use profiling profiles of tumors and liquid, exRNA, with an idea that we want to bridge the two, right? We talked a lot about liquid biopsy biomarkers. Our ambition is to deconvolute both heterotypic interaction within tumors and then exRNA that leaks into accessible biofluids. So we pursue quadratic programming, matrix factorization approach. So basically, you have measured transcript levels, columns are samples, and rows are transcripts on the matrix on the left. You want to represent it as a product of two relatively thin matrices. So the green one should have few columns. They correspond to different cell types, and few rows. The number of columns and rows in the green and rows in the blue are the same. And the blue one contains the proportions. When it comes to exRNA, the problem is the same, except we're not talking about bulk RNA profiles of tumors, but exRNA profiles. And what we want is to infer both the number of different cargo types corresponding to different carriers and the proportion of each cargo profile in individual samples. So I discussed this, brought up this issue yesterday. There are actually two types of deconvolution methods, reference-free and reference-based. So reference-based deconvolution we heard about yesterday from Rob Kitchen and others assume that the green matrix is given, right? Known. We know what are the profiles of different carriers. We are just estimating the blue matrix, whereas in reference-free deconvolution, we are estimating both the green and the blue matrix simultaneously, and not only that, but we are estimating actually a number of columns in the green matrix and the number of rows in the blue matrix. So what I'll be talking today is reference-free deconvolution in contrast to reference-based, which we heard about yesterday. In either case, both for reference-based and reference-free deconvolution, informativeness of features is very important. So we need the transcripts whose levels varies across different cell types or across different carriers, because otherwise we don't have the information. So you can think about this factorization being really an attempt to explain the variance in observed transcript levels by decomposing it into variance due to relative abundances and changes with individual carriers. So exDec process has two stages. In stage 0-1, only a subset of most informative transcripts is used to essentially estimate the blue matrix and fix it for stage 2. Now, green and blue are estimated simultaneously, even in stage 0-1, except because of scaling, which I'll talk about shortly. In stage 2, actually, the final version of the green matrix, which includes all transcripts, is computed. So as I mentioned, the deconvolution algorithm is an iterative algorithm for constrained matrix factorization using quadratic programming. A mouthful for you who are not mathematically inclined, but basically it is essentially a least square fitting method. It generalizes multiple regressions. So if you think about green matrix being fixed as in reference-based deconvolution, we're talking about something very similar to multiple regression, right? We're just computing the Wu matrix. But, and in fact, one can use, multiple regression algorithms. But if both the green and blue are estimated, then this is real, then you need this constrained matrix factorization using quadratic programming to solve this optimization problem. So here's the whole process. So stage 0 is really a selection on informative loci based on single cell RNA-seq profiles in case of tumor deconvolution, or isolated exRNA-seq profiles from isolated carriers in case of exRNA. In stage 1, which is the middle one, there is this iterative cycle where V is approximated as a product of W and H matrices, least squares optimization essentially. And then once the proportions are fixed in stage 1, then in stage 2, one can actually deconvolute any other omic layer, say RNA-seq layer or even proteomic layer, bulk profile, using essentially multiple regression or something similar to reference-based deconvolution. So here's an example in preparation where we applied this to TCGA breast cancer tumors. One can identify 9 different cell types. So because they add up to 1, there's actually 8 degrees of freedom, 8 dimensions to this data, which can be visualized as 8-dimensional simplex. And we actually can reproduce the same cell types both from methylation, bulk methylation deconvolution, and RNA-seq based. You can see cancer such as fibrous adipocyte axis, and then you have this triangle of different cancer subtypes. There are 3 main ones in breast cancer, which actually we could reproduce by deconvolution: luminal, HER2, and basal. You see the projection here of the stroma, which is goes from yellow on the left to gray onto the subtype of breast cancer, luminal in the bottom left, HER2 positive in the bottom right, and basal in the top. So you can see, you can observe correlations with different stroma and epithelial types, which actually are due to exosome exchange. Stromal glycolytic cells secrete lactate-rich exosomes into HER2 cells and upregulate their metabolism. We actually deconvoluted glioma as well, published about a year ago. Identified the role of exosomal miR9-5P in angiogenesis. These are miRs secreted by cancer cells promoting angiogenesis by a pathway that's parallel to a growth factor pathway. And miR9P is also elevated in plasma of glioblastoma patients, allowing us to now hypothesize that we can have a liquid biopsy biomarker by deconvoluting both the tumors and the exRNAs. Now, our main application was exRNA atlas deconvolution, where we deconvoluted all the data in the atlas 2 years ago. What was important is the same fractions were deconvoluted, cargo types, which we call cargo types, CT1, CT2, 3A, BC, and CT4, from different studies using different biofluids, as indicated on the right. And they clustered together. This led to hypothesis that they correspond to different carriers. And in fact, this was validated, karyotype 1 being linked to one class of vesicle, CT2 to lipoproteins, by correlation of deconvoluted profiles with isolated exRNA profiles, say from HDL and LDL and VLDL carriers. CT3, A, B, and C are RNP particles. as evidenced by correlation with AGO2 pull downs and profiling them by our collaborators. And then CT4 is another vesicle class. We also show that deconvolution helps in case control studies by increasing the power of case control study by separating variants. Variance is just carrier abundance, variation in individual samples versus variations in the cargo within individual carriers of exRNAs. So using a previously published study in 2017 that involved 26 pre and 36 post-exercise plasma exRNA samples, we deconvoluted into 3 cargo profiles as indicated on the right, HDL, RNP particles and extracellular vesicles, and observed changes upon exercise, increase of EVs relative to RNP particle and HDL cargo. And then looking at inside, changing composition, we found relevant pathways based on microRNA enrichment, all relevant to muscle contraction and metabolism changes. Now, is this just an artifact of secretion, or do these microRNAs regulate target tissues? This is still open, but at least we've got a physiologically plausible signal, which was not obtained in the previous study because it didn't use deconvolution. So before we close, I just wanted to mention 2 major sources of biases that impact deconvolution. One is the RNA extraction kits. They have biases towards specific exRNA carriers, vesicles, RNP particles, lipoproteins, and library preparation kits, which is a different story. This is has to do with this adapter ligation, and we've heard yesterday about covalent modifications and how they make certain, say, ribosomal RNA fragments invisible to sequencing because they're just not, phosphorylated appropriately to be captured in these by these adapters. So here's the analysis of 10 different RNA extraction kits. As you can see, as we deconvolute their products, we see that some of them isolate EVs of one type, another of another type, and some of them, in fact, enrich for RNP particles, not EVs at all. In terms of library preparation, that's a serious problem, actually more complicated than RNA-seq of tumor profiles, because there is this library preparation may lead to huge over or under sampling of specific transcripts by orders of magnitude. And these biases may change from one version of the kit to another. These companies improve kits and change performance unbeknownst to the users. So different subsets of transcripts consequently may be visible or sampled with any given kit or version of the kit. So selection of informative transcripts may not be valid from one study to another. And these biases may also cause loss of reproducibility of deconvolution, right? So if you have a finding and then use a different kit, we may not be able to reproduce our findings. So our solution to this problem is scaling into 0-1 range, right? Instead of dealing with uncorrected transcription levels, all the expression levels of RNA-seq profiles are transformed prior to deconvolution, and then only in the last step, the non-scaled RNA-seq quantities are used to deconvolute. So in summary, I've shown that feature selection is important for reference-free deconvolution. Single-cell RNA-seq profiles help identify important features for tumor deconvolution, isolated exRNA carriers for exRNA. And they also help validate results of reference-free deconvolution by identifying deconvoluted fractions by correlation. Scale invariance is very important in reproducibility, especially for exRNA data where there's huge variability in transcript levels, even from one version of the kit to another. But despite all of that, we've shown that exotic deconvolution reveals biases of RNA extraction kits. We can see what they sample. We can identify carrier-specific cargo profiles across different studies, extract potentially physiological signals from case control studies by separating variants, and link paracrine signaling that's microRNA mediated with potential exRNA biomarkers. So with this, I'd like to thank Oscar Murillo, Emil LaPlante, Raduhi, Petrosian, who are now working on the exec paper, Rocco Lucera, who did Glioblastoma, Matt Roth, who participated in all these studies, Michael Lewis, our collaborator at Baylor, who does PDX modeling and breast cancer, and many ERCC collaborators. I could fill the screen, but so that to just say many of you have participated. Thank you.
Thanks a lot, Alex. We have a brief moment for questions. Someone on onlinequestions.org asks, how does reference-free deconvolution compare to other factorization approaches such as surrogate variable analysis?
Surrogate variable analysis, or SVA, also stands for singular value analysis, singular, or SVD, singular value decomposition, non-negative matrix factorization. There's a number of these methods that are very similar. We actually, as part of a bake-off of epigenomic deconvolution methods, we have something similar to the workshop we are considering now, but aimed at DNA methylation data, right, which is kind of already scaled into 0-1 range. We looked at these different methods that combine these ideas, and my conclusion is that there is the deep underlying structure of all these methods is very similar. And actually, performance of all the epigenomic deconvolution methods was dependent on a similar number of factors. So if the transcripts were informative, you would get similar results using these different methods. So yeah, so without going into details, you know, they all exploit similar structure in the data, which can be expressed as matrix factorization.